So today we'll be solving May-June 2024 paper 1 variant 1. सबसे latest paper. Let's see. And काफी easy paper है ये. Question number 1. The diagram shows a stage micrometer scale viewed with an eyepiece graticule using a magnification of 200. Using the same magnification, a chloroplast is measured as 4 eyepiece graticule division long. How long is a chloroplast? अच्छा हमें पता है to calibrate your eyepiece graticule, we do not need to look at the magnification. So you're going to ignore the statement using a magnification of 200 micrometer. 200, sorry. अच्छा. First, identify stage micrometer कौन सा है and eyepiece graticule कौन सा है. So this one is your stage micrometer and this small one is your eyepiece graticule. Your stage micrometer calibrates your eyepiece graticule. So in 0.1 millimeter, we can see 90 minus 50 is equal to 40 eyepiece graticule unit. सारे आंसर्स आपके माइक्रोमीटर में है, सो विल कन्वर्ट 1 मिलीमीटर इनटू माइक्रोमीटर. 0.1 millimeter is equal to dash micrometer. We know 1 millimeter is equal to 1000 micrometer. So simply cross multiply करेंगे and our answer will come out to be 100 micrometer. From this 0.1 millimeter वाली बात कर रही हो? नहीं नहीं, ये तो आपके क्वेश्चन पे डिपेंड करता है. Usually 0.1 millimeter is equal to 10 eyepiece graticule. 100 eyepiece graticule लिखा होता है क्वेश्चन में. कभी कभी 0.2 मिलीमीटर भी हो सकता है. दिस डिपेंड्स ऑन योर क्वेश्चन. यहाँ पे मैं डायग्राम में इन्होंने लिखा हुआ है कि 0.1 मिलीमीटर इज इक्वल टू इतना eyepiece graticule. 90 minus 50 comes out to be 40 eyepiece graticule. So in a way we can say 40 eyepiece graticules are equal to 100 micrometer. Now you need to calibrate one eyepiece graticule कितना मिलीमीटर माइक्रोमीटर होता है. 1 EPG is equal to x. क्रॉस मल्टीप्लाई करेंगे. x is going to 100 divided by 40 and your answer is 2.5 micrometer. 1 eyepiece graticule is equal to 2.5 micrometer. ये हमने कैलिब्रेट कर लिया आपका eyepiece graticule from your stage micrometer. Now they have given in the question कि your chloroplast is measured as a 4 eyepiece graticule unit. So simply we're going to cross multiply again. 4 eyepiece graticule is equal to x micrometer. Cross multiply 4 multiplied by 2.5. Answer is 10 micrometer. पावर में लिखेंगे कैसे लिखेंगे? 1 into 10 की पावर 1 micrometer. एक ही बात है. So correct answer is A. सही है? Is this clear? Yes, 3 steps. पहले आपने आइडेंटिफाई किया कि स्टेज माइक्रोमीटर वाली बात, दस कन्वर्जन इनटू माइक्रोमीटर, मिलीमीटर टू माइक्रोमीटर, बिकॉज आपके सारे ऑप्शंस माइक्रोमीटर में थे. आप एन में भी कन्वर्ट कर सकते हो बट कंफ्यूजन हो सकती है इन कैलकुलेशन. तो बेटर है कि आप स्टार्ट में कन्वर्ट कर दो बाय लुकिंग एट योर ऑप्शन. फिर यू कैलिब्रेटेड योर आईपीस ग्रेटिक्यूल. दिस इज द कैलिब्रेशन ऑफ योर आईपीस ग्रेटिक्यूल. थर्ड स्टेप वाज टू फाइनली कैलकुलेट कि कितना मिलीमीटर होगा, सॉरी माइक्रोमीटर होगा. सो दिस वाज द थर्ड स्टेप. Question number okay. Question number 2. Which range of cell diameter is typical for prokaryotic cells? This is a learning point and your prokaryotic cells are in the range of 1 to 5 micrometer. If you look at your options, इन्होंने nanometer में भी दिया हुआ है. So for our convenience, let's convert it into nanometer as well. So 1 nanometer is equal to, sorry, 1 micrometer is equal to 10 की power 3 nanometer. So we can say 1 into 10 की power 3 nanometer से लेके 5 into 10 की power 3 nanometer, ये भी हो सकती है conversion. Let's look at the option. 1 nanometer, this is wrong, 1000 होना चाहिए था, so option 1 is wrong. 1 into 10 की power 3 nanometer to 5 micrometer, this is exactly correct. तो बाकी option obviously are wrong. And आपको नजर भी आ जाएगा mistake कौन सी है. Question number 3. The diagram shows a typical animal cell. Let's identify. One is your, one क्या नजर आ रहा है हमें? Yes, one is your cytoplasm. 2 is your, if I zoom in, Yes, ribosome. Yes. Ribosomes on your aria. Ribosomes on aria. And three is your mitochondria. Hmm. Mitochondria. Question says, where would nucleic acid be found? So काफी easy सा question है. What do you think would be the answer? Ribosomes में you have ribosomal RNA. This is a type of your nucleic acid. Mitochondria में you have circular DNA. So you have all types of DNA and RNA in your mitochondria. Cytoplasm, DNA and RNA. Cytoplasm में you have mRNA, you have ribosomes. So hence obviously cytoplasm maybe you have nucleic acid. Correct option है तीनों ही होंगे 1, 2 and 3. माइटोकॉन्ड्रिया का अपना सर्कुलर डीएनए होता है and DNA is made of nucleic acid. So all three of them are correct. Which structure is found in a typical bacterial cell? Introns are not found in your bacterial cell. Telomere is present in linear DNA only. Telomere की presence के लिए there is a condition. There should be ends on your DNA molecule. Linear DNA में you have two ends, but whereas in your circular DNA there are no ends. तो telomere की need ही नहीं होती in your circular DNA. So always remember telomeres are always only found in your linear DNA. Needs ends. And bacterial cell, you know, it has circular DNA. Template strand. This is found in your bacterial cell क्योंकि it has its own circular DNA. So template strand found होगी capsid. Capsid is present in your virus only. According to your course, it is present in virus only. So correct option is C. Question 5 is इसमें थोड़ी सी mistake है. What is the maximum number of hydrogen bond that can form between a single water molecule and other water molecule? Now, पहले जब हमने ये क्वेश्चन किया था तब इनका आंसर था 1. दिस इज अ रिपीटिंग क्वेश्चन लेकिन हर बार क्वेश्चन का आंसर डिफरेंट आ रहा होता है. Sometimes it's 1, sometimes it's 2 and sometimes it's 4. According to this paper your answer is 4 and वैसे भी 4 ही होता है आंसर. 1 and 2 are not the correct option. किस तरह होता है? Let me show you the structure. एंड ये आपने केमिस्ट्री में भी पढ़ा होगा. यस 4 ही बनता है बट एक और पास पेपर मैं कर रही थी तो उसमें 2 था एंड फिर थर्ड वाले पास पेपर में इट वाज 1. तो या तो मार्किंग स्कीम गलत थी या सीआई वाला कंफ्यूज था बट वी नो 4 बनता है. सो यू आर गोइंग टू मार्क 4. This is your partial positive hydrogen atom. This is your partial negative oxygen atom with two lone pairs of electron. Each lone pair will form one hydrogen bond. Oxygen के each lone pair will be forming one hydrogen bond and hydrogen itself will be forming hydrogen bond with other hydrogen molecules. सही है? Other water molecules. So which is why it will form four hydrogen bonds. Question 6 is very interesting. The diagram shows the structure of four amino acids in aqueous solution. Which two structure have an overall charge? The structure 1 is of your glycine, एक negative, एक positive. It is going to cancel itself out. Cancel out हो गया, there is no charge on glycine. Alanine, एक negative, एक positive, again cancel out हो गए. Lysine, एक नेगेटिव एक पॉजिटिव दिस इज गोइंग टू कैंसिल इटसेल्फ आउट एंड फिर एक यहाँ पे एक और पॉजिटिव है. सो ओवरऑल देयर इज अ पॉजिटिव चार्ज ऑन योर लाइसिन मॉलिक्यूल हेंस वी कैन से योर लाइसिन इज पॉजिटिवली चार्ज. फिर यू हैव एस्पार्टेट. इसमें एक माइनस एक पॉजिटिव अगेन कैंसिल आउट बट देयर इज ऑल्सो अ नेगेटिव चार्ज एक्स्ट्रा एंड वही रहा. सो दिस इज नेगेटिवली चार्ज. So here, according to this, these two structures are charged. Overall charge, they are carrying an overall charge on their structures. Correct option is lysine and aspartate. C. ऐसा क्वेश्चन आपको केमिस्ट्री में भी आता है. इलेक्ट्रोफोरेसिस में आता है आई थिंक. क्वेश्चन 7 थोड़ा सा ट्रिकी है but काफी interesting question. The table shows the number of carbon atoms and the number of carbon-carbon double bond in molecules of three triglyceride and the fatty acids they contain. You have three types of triglyceride एंड उन दोनों के तीनों के फैटी एसिड नंबर दिए हुए इन्होंने. द इंफॉर्मेशन इज शोन इन द फॉर्मेट number of carbon atoms and number of carbon-carbon double bond. So you know triglyceride is made up of fatty acid and glycerol. They've only mentioned fatty acid, glycerol इन्होंने नहीं दिया होगा, but you should know the structure of your glycerol. याद कर लो. Glycerol has 3 OH in its structure and it looks like this. And this information will be applied in this question. Let's see. थोड़ा सा मैं इसको साइड पे कर देती हूँ. Which description matches fatty acid 2 of the 56,4 triglyceride identified in the table as X? अच्छा, ये रहा आपका triglyceride. This is your fatty acid 2, fatty acid 3. Ab, how is this 54 number coming out? If you look at this, 15 plus 18 plus 18 gives you, एक का करके दिखा रही हूँ, 15 plus 18 plus 18 gives you 51. Acha. And this is 54. If you look at your glycerol, you have 3 carbon atoms in your glycerol. So 51 plus 3 from your glycerol will give you 54. This is how there are 54 carbon atoms in your triglyceride 1. This is your triglyceride number 1, triglyceride 2, triglyceride 3. फिर this one is for your number of carbon-carbon double bond. So वो ये रहा in your fatty acid number 3. इस तरह आपका ये रेशो बन रहा है. Same method of calculation for 2 and for 3 as well. 3 को देख लेते हैं, फिर we'll move on towards 2, जैसे हम अपना आंसर कैलकुलेट करेंगे. So 18 plus 18 plus 18 is 54. 54 and 3 carbon atoms from your glycerol molecule. 54 plus 3 is equal to 57 total. And number of carbon-carbon double bond वो आप डायरेक्ट यहाँ से निकाल सकते हो 2 and 1 which is 3. ये रहा. Same method for your carbon triglyceride 2. 17 plus x plus 18. अच्छा, पहले आप क्या करोगे, थोड़ा सा आप इसको बैकवर्ड्स करोगे. 56 minus 3. This is going to give you the number of fatty acid के carbon. 56 minus 3 is 53. तो यहाँ पे जो भी आपका सम आएगा that should be equal to 53. So किस तरह करेंगे, 17 plus x plus 18 is equal to 53. x आपका आएगा 18. So your fatty acid number 2 will have 18 carbon. डबल बॉन्ड कितने होंगे, 4. यहाँ पे 2, ऑब्वियसली यहाँ पे 2 होंगे. यहाँ पे 0 है fatty acid 1 में, तो ऑब्वियसली बाकी डबल बॉन्ड will be in your fatty acid number 2. थोड़ा सा मैथ वाला क्वेश्चन है ये, but for this you should know the structure of your glycerol. 3 carbons are coming from your glycerol. ये रहा स्ट्रक्चर, सब में यही हो रहा है. अच्छा एक मिनट, बल्कि दिखाई देती हूँ. इफ़ यू कैलकुलेट 15, 18 एंड 18 प्लस करोगे क्या आएगा नंबर, कैलकुलेटर निकालो. 51 आ रहा है, बट यहाँ पे क्या लिखा हुआ है, 54 लिखा हुआ है, विच मीन्स कि बाकी जो 3 कार्बन है दोज आर कमिंग फ्रॉम ए ग्लाइसरॉल का मॉलिक्यूल. एंड वी नो ट्राइग्लिसराइड इज मेड अप ऑफ 3 फैटी एसिड, 3 फैटी एसिड्स एंड 1 मॉलिक्यूल ऑफ ग्लाइसरॉल. इन्होंने यहाँ पे सिर्फ फैटी एसिड का मेंशन किया हुआ है. ग्लाइसरॉल आपकी नॉलेज होगी. इज दिस क्लियर? तो ये थोड़ा सा है कंफ्यूजिंग क्वेश्चन. अच्छा, सिंस इट हैज़ अ डबल बॉन्ड, यहाँ पे हमने देखा 2 डबल बॉन्ड, it will be a unsaturated fatty acid with 18 carbon atoms. Option D is your correct answer. So अगर आपको glycerol का structure नहीं पता होता, you wouldn't have been able to solve this question. Question 8. Collagen molecules are made up of 3 polypeptide chain interacting together. कैसे interact करते हैं? By your hydrogen bonding. The individual polypeptide chain consists of a regular pattern of amino acid. Almost every third amino acid is glycine. Which protein structure of collagen are described? इसमें which protein structure of a collagen molecule are described? So let's see. Let's start to highlight your keyword. Regular pattern of amino acid. मतलब sequence of your amino acid, and this is referring to your primary structure. 3 polypeptide chain. 1 or more polypeptide chain. This is referring to your quaternary structure. So they are talking about your primary and quaternary structure. Why not secondary? Secondary is made up of alpha helix and beta pleated sheet. वो मेंशन ही नहीं किया यहाँ पे इन्होंने, अल्फा हेलिक्स एंड बीटा प्लीटेड शीट, so hence it cannot be your secondary structure. Correct answer is B. Is this clear? अच्छा, इस पेपर में काफ़ी मैथ वाले क्वेश्चंस हैं, so make sure you remember the formulas for percentage increase and percentage decrease. Question number 9 is also calculation वाला question. The masses of the part of hemoglobin are shown in the table. Component दिया हुआ है and उसका mass दिया हुआ है in Dalton. Alpha globin chain का weight is this in Dalton, beta globin chain and your heme group. There are 1000 Dalton in 1 kilo Dalton. What is the mass of hemoglobin molecule in kilo Dalton? To solve this question, you should know the structure of your hemoglobin molecule. किस तरह दिखता है? It has two alpha globin chain, two beta globin chain, and four heme groups. Four heme groups. कुछ भी नहीं करना आपको, आपको सिर्फ मल्टीप्लाई करना है इन वैल्यूज को एंड देन कैलकुलेट करना है सम. So one alpha globin chain का mass is 15126 multiplied by 2. The mass will come out to be 3256 Dalton. Same for a beta globin chain. 2 होती हैं तो multiply by 2. Mass will come out to be 31736 Dalton. One heme group is of 617 Dalton. 4 heme group कितनी होंगी? Multiply by 4. 2468. Now calculate the total mass ताकि आपको पता चले the mass of a hemoglobin molecule. हल्की सी आपकी नॉलेज लगी, बाकी मैथ स्किल जो आप कैलकुलेटर से कर सकते हो. दिस टोटल विल कम आउट टू बी, इन तीनों वैल्यूज को ऐड करना एंड इट विल कम आउट टू बी 64456 Dalton. We need to give our answer in kilo Dalton. 1 Dalton, sorry, 1 kilo Dalton is equal to 1000 Dalton. X kilo Dalton in 64456 Dalton. Cross multiply किया, divide by 1000 किया, and your answer will be 64.5 Dalton. Okay, एक minute. 64.456 Dalton. Answers are in one decimal place. आपने इसको round off किया, 64.5 Dalton. This is your answer. वैसे तो आंसर यहीं पे पता चल रहा था एंड डायरेक्टली आप कर लेते, बट मैंने शो कर दिया एक्स्ट्रा वर्किंग. योर आंसर इज डी. इज दिस क्वेश्चन क्लियर? Okay. What is the correct order of location in the cell for the production of extracellular enzyme? This is a repeating question. Yes, Sana? They can replace the word enzyme by glycoprotein. They can replace the word enzyme या फिर glycoprotein by mucus as well. So तीनों का method same है. Location and production का method बिल्कुल same होता है. It starts from your nucleus and it ends with your Golgi body. तो end पे आपका होना चाहिए Golgi body, start पे आपका होना चाहिए nucleus. A is your correct answer. किस तरह होता है? From your nucleus, your primary transcript या फिर your mRNA is released. mRNA attaches to your ribosome. Ribosomes are present on your rough endoplasmic reticulum. फिर vesicles बनती हैं. Those vesicles go to your Golgi body for further um, translational modification, further protein modification basically. And फिर Golgi body से आपकी vesicle निकलती है. Vesicle goes towards the cell surface membrane. Exocytosis का process होता है. अगर इसको further करना हो, vesicles goes towards your cell surface membrane. Fusion होती है, exocytosis होता है, and your enzyme, your glycoprotein or your mucus is released. अगर mucus की बात होगी तो goblet cells होंगे. And you know mucus is a type of glycoprotein. तो एक ही बात हो गई. सही है? Is this clear? Okay. So always remember protein synthesis आपका हमेशा nucleus से start होता है, not from your ribosomes या फिर rough endoplasmic reticulum. Question 11. Question 11 is very easy. This is your homework. I think एक ही homework होगा आज. Which statement about the KM are correct? The higher the Km, the higher the enzyme affinity for the substrate. This is wrong. बिल्कुल inversely proportional होते हैं. The higher the Km, the lower the enzyme affinity for its substrate. One is wrong. Correct answer is D. But let's see the option. Km is a measure of the degree of enzyme affinity for a substrate. This is correct. Km is defined as a substrate concentration at which the enzyme functions at half its maximum rate. They're talking about Vmax over 2. तो बिल्कुल exactly यही हो रहा होता है जो Km is calculated जहाँ पे आपका Vmax half हो रहा होता है. Vmax half हो रहा होता है on your y axis. फिर आप corresponding value निकालते हो from your x axis. So option number 3, statement 3 is also correct. D is your correct answer. काफी obvious answer था. An elimination method भी हो रहा था. Question 13. The diagram shows how nicotine is transported from the blood plasma into a cell using a type of co-transporter mechanism. Acha, let's see. Nicotine is transported against the concentration gradient, matlab ATP use हो रही है and this is an active transport का process. Whereas आपके जो protons हैं, they are moved out of your cell down the concentration gradient. Diffusion हो रही है, facilitated diffusion हो रही है, and it is a passive process, no need for ATP. Acha. Your protons are pumped out into your blood plasma, whereas nicotine is pumped into the cytoplasm of your cell. ये सब हमने study कर लिया from your diagram. In the phloem tissue, there is a co-transport mechanism that moves sucrose into the cytoplasm of a cell. This is true. ये हमने देखा भी in the process of translocation. But the difference is जो आपका phloem tissue में जो आपका transport protein होता है, co-transport protein होता है, that is pumping sucrose as well as proton into a cell. यहाँ पे opposite हो रहा है, एक अंदर जा रहा है, एक बाहर जा रहा है. So remember, sucrose अंदर जा रहा होता है. By the process of active transport यहाँ पे ATP required होती है and it is moving against the concentration gradient. Whereas your protons, आपके cell में protons कम होते हैं. So they're moving down the concentration gradient by the process of diffusion, facilitated diffusion, and there's no need for ATP. Which statement correctly describes a similarity between the co-transport of nicotine and the co-transport of sucrose? The co-transport protein generate a proton gradient by moving proton out of the cell. No, they're moving proton into the cell. Your co-transport protein in phloem tissue, they're moving your protons into the cell. ये हमने देखा. Let's say this is your blood plasma. I think थोड़ा सा नीचे हो गया, ऊपर हो गया. एक मिनट. This is your blood plasma. Sorry. Phloem tissue है. So this is your companion cell के अंदर जा रहा होता है आपका. And phloem sieve tube या कहीं से भी बाहर से आपका अंदर आ रहा होता है. So this is the protons are pumped into your cell. And they're pumped by passive process, passive transport. So pura sentence is wrong. The protons are transported through the co-transport protein by facilitated diffusion. इस सेंटेंस में क्या इशू है? कोई भी इशू नहीं है. बिल्कुल करेक्ट लिखा है इन्होंने. एंड यहाँ पे सक्रोज की मूवमेंट भी हो रही होती है. तो दिस सेंटेंस इज करेक्ट. द प्रोटॉन्स मूव through the co-transport protein in the opposite direction to the movement of nicotine and sucrose. The protons move through the co-transport in the opposite, in the same direction as the sucrose and in the same direction as the nicotine. सेल के अंदर जा रहा है आपका निकोटीन तो बिल्कुल सेम डायरेक्शन होता है. So this sentence is also wrong. Same होना चाहिए था. The co-transport protein use energy from ATP to transport proton. Your protons are transported passively. So there's no need for ATP. With nicotine and sucrose, this is wrong. यहाँ पर भी diffusion हो रही है. So again, nicotine वाला point भी wrong है, sucrose वाला point भी wrong है. Correct option is B only. अच्छा, they are only looking at your proton. Sucrose की movement नहीं देख रहे. So yes, B is your only correct answer. Is this clear? Okay. The diagram shows a simple metabolic pathway. W is converted into X, Y, and Z. The letter W, Y, X, Y, and Z represent four different substances. At each step in the diagram, the substrate undergoes a chemical reaction catalyzed by an enzyme. The reaction produces the next substance in the pathway. Acha, let's see which statement correctly describes the enzyme taking part in this metabolic pathway. So you're going to look at the properties of your enzyme. Recall the properties of your enzyme. They're all globular protein. This is true. All your enzymes in your cell, in your body, are globular protein. None of them are fibrous protein. They all have the same tertiary structure. अगर सबका tertiary structure same होता तो सबका substrate भी same होता. Which is why this is statement is logically wrong. They have different tertiary structure. Different tertiary structure. They all contain hydrogen atom in their structure. अच्छा. Enzymes are made up of protein. Proteins are made up of amino acid. And amino acid का structure कैसा होता है? Amine group, carbon, carboxyl group, ऊपर hydrogen, and then there's an R group. So there is always an hydrogen atom in your protein, your enzyme. ये रहा hydrogen, ये रहा, and ये रहा. So they all contain hydrogen atom in their structure. This is true. All your enzyme contains hydrogen atom in their structure. Option 1 and 3 only. C. C is the correct answer. Is this clear? Okay. काफी easy paper बना है ये थोड़ा सा confusing होगा. A student observed the effect of two different concentration of salt solution on blood cells. Okay, blood cell, so either it will shrink या फिर burst. The student added each concentration of salt solution to one of two microscopic slide and then a small drop of fresh blood was added. सही है. Microscope slide पे you put salt solution फिर आपने अपना blood डाला. Each slide was viewed using a high power lens of a microscope and the student's observation were recorded. No red blood cells were visible. The red blood cells were visible but looked slightly crinkled. Biconcave shape होता है and they become crinkled. So obviously there is a shrinking in your cell. आपका cell shrink हो गया है. Shrink. No red blood cells were visible, so this means कि आपका cell burst हो गया, which is why you were unable to see any red blood cells burst हो गया. Which row correctly explains the result obtained? Now this sign, this symbol is for your water potential. Water potential का symbol इस तरह होता है. की भी दी हुई है इन्होंने. ये कोर्स पहले होता था आपके कोर्स में, but I don't think अब है. अच्छा, if you look at your options, more negative, less negative. आप इसको पढ़ते पढ़ते भूल जाओगे. More negative क्या होता है, less negative क्या होता है. So for your easyability, की के साथ ही आप अपनी एक और की बना लो. More negative means low water potential. More positive means high water potential. तो एक छोटी सी की अपनी बना लेना before solving this question, वरना आप confuse हो जाओगे. Swelling of the cells cause them to burst. Side 1 में यही हो रहा था, your cells were bursting. ये हमने conclude कर लिया था from the observation by the student. The water potential of the cell was more negative, which means high, was more negative than the water potential of the external solution. More negative means low, but shrinking हो रही है. So your water moved from a region of high potential to a low potential, which was outside external solution. अच्छा, more negative भी लिख देते हैं यहाँ पे. If more positive, more negative is low. अच्छा लिखा, sorry. The water potential of the cell was more negative, which is low water potential, than the water potential of the external solution. This is completely opposite. Shrinking हो रही है, what is moving out of your cell? Water moves from a region of high water potential to a region of low water potential, which is outside your cell. यहाँ पे बिल्कुल opposite हो रहा है. Slide number 2 की observation is wrong. The water potential of the cell was more negative than the water potential of the external solution. More negative, low water potential, then the water potential of the external solution मतलब water entered your cell, जिसकी वजह से your cell eventually bursted. So this observation is correct. The water potential of the external solution was more negative, again low, but they're talking about your external solution this time, which is correct. Yes, जिसकी वजह से water left your cell and entered your solution, and then eventually your cell shrinked. This is correct. So option B is going to be your correct answer. And as you saw, मैं खुद भी confused हो रही थी. So it's better if you guys make a key for this question के साथ ही कि more negative क्या होता है, more positive क्या होता है. And वो मैंने यहाँ पे mention कर दिया. You can learn this as well. Is it clear? Okay. Which statement correctly describes facilitated diffusion? Facilitated diffusion happens to your transport protein. There are two types of transport protein, which are your carrier protein and your channel protein. So let's see the statements. The process only occurs using channel protein. It occurs through a carrier protein as well. So ये शुरू में गलत हो गया. That change shape and that uses energy provided by the cell. अच्छा. Your carrier protein may or may not use ATP and it may or may not. अच्छा. It does change shape. Does change shape. Your carrier protein. But as a channel protein, there is no need for changing in shape. Hence, there is no need for ATP. No ATP needed. सबसे common type of your channel protein is your aquaporin, the ones which are transporting your water molecules across the cell. So आगे वाली pointer भी wrong है. Your channel proteins are never changing shape. एक channel बना हुआ होता है and water molecule आराम से आपका transport हो जाता है. And it is not using it energy provided by the cell. ये दोनों बातें आपकी wrong है. तीनों बातें wrong. The process occurs using channel protein or carrier protein that may or may not change shape. This is true. The channel protein is not changing shape, but your carrier protein, according to your course, is changing shape. वैसे there are certain carrier protein which do not change their shape, but आपके course के हिसाब से they are changing their shape. The process occurs using channel protein or carrier protein that uses energy provided by the cell. The mistake is that may or may not use energy. May or may not use energy by the cell. Your channel protein is not using any energy. The process only occurs using carrier protein. The process occurs through your channel protein as well. So option C and D are also wrong. ये भी थोड़ा easy था. Which row is correct for stem cell? Can repair cell. Always remember, आपकी body में कोई भी ऐसा cell नहीं है which can repair another cell. या कोई भी ऐसा process नहीं है which can repair another cell. Your cell, once destroyed, can only be replaced. Mitosis भी आपका cell replace करता है. It is not repairing your body cell. So stem cells cannot repair your cell. Can be involved in the formation of phagocytes. Phagocytes क्या होते हैं? They are a type of white blood cells. Stem cells can differentiate into white blood cells. Your hmm, yes, बिल्कुल सही. Your stem cells can differentiate into white blood cells, your red blood cells as well. आपके fat cells, neuron cells, body के हर type of cells में ये differentiate कर सकते हैं. And we know white blood cells की एक type होती है phagocyte. So this is correct. यहाँ पे yes होना चाहिए. Correct option is D. Eukaryote grow and increase in size as a result of cell division. The diagram shows some of the stages of mitosis with one stage missing labeled X. What happens in stage X? So mitosis में P-mat हो रहा होता है. Prophase, metaphase, anaphase and telophase. Your spindle fiber, your chromosomes, sorry, are arranged along the equator of the cell. And this happens during your metaphase. So बीच में you have anaphase and this is obviously your telophase. So they are missing anaphase. You're going to look for the properties of anaphase in your option. Spindle fiber attached to the centromere and the chromosome line up along the equator. ये हमें पहली वाली diagram में नजर आ रहा है. This happens during your metaphase, so this option is wrong. The chromatin coils and condenses, and the nuclear membrane disintegrates. This happens in your prophase. You want to look for the statement with anaphase feature. The chromosome line up in pairs along the equator of the cell. Again, वही बात, this is happening during your metaphase. Microtubules shorten so that spindle fiber contract and the centromere divide. This is what is happening during your anaphase. वैसे आपको बाकी स्टेटमेंट देख के भी इजीली आइडेंटिफाई हो जाता है कौन से फेज में क्या हो रहा है. क्वेश्चन 19. ये भी easy है. The graph shows various distances. Distance measurement taken from the start of the metaphase of mitosis. The graph are to scale when compared to one another. अच्छा. Distance आपका पहले constant था and then started to decrease. So इस graph में your distance is decreasing. Decreasing. इस ग्राफ में क्या हो रहा है? If you look at the gradient of your graph, maximum फिर decrease होता जा रहा है gradient. So in this graph as well, the distance is decreasing. थोड़ी सी मैथ्स वाली नॉलेज या फिर फिजिक्स वाली एक ही बात. अच्छा, which who correctly identifies the distance measurement for each graph? Which phase was this? This is the metaphase. Metaphase में क्या हो रहा होता है? आपके जो chromosomes होते हैं, they are aligned along the equator of your cell. कुछ इस तरह नजर आता है. This is your cell. यहाँ से आपके spindle fibers आते हैं from your centrioles. This as well. And ये रहा आपका chromosome along the equator of your cell. Your spindle fiber it attaches to the centromere of your chromosome ताकि वहीं से आपका पुल कर सके आपका क्रोमोसोम को पुल कर सके towards the cell. Which you correctly identifies the distance measurement for each graph. Distance between the poles of spindle. Distance between the poles of spindle. वो तो constant ही रहता है, change नहीं हो रहा होता. This is wrong. Distance of centromeres from the poles of spindle. Distance of centromeres. Since your centromeres are going towards the pole of your cell, the distance obviously decrease कर रहा है. Are you able to understand this? किस तरह decrease कर रहा है? Your spindle fiber will contract and pull your centromere towards itself. So obviously this distance is going to decrease. Distance between your centromere and your poles of spindle. ये decrease होता जाएगा eventually. So this is x. X वाला ग्राफ है ये. Y. Y क्या होगा? Y में भी आपका distance decrease कर रहा है. Distance of centromere from the poles of spindle. Distance between sister chromatid. Distance between sister chromatid and distance between poles of spindle. Distance between sister chromatid. इसको क्या होगा? आपका centromere divide करेगा and it will start to move towards the opposite pole. So distance between your sister chromatid will increase. यहाँ पे increase आएगा. But your graph is showing a decrease in distance. Hence, your correct option is D. Is this there? Next one. Moving towards the opposite pole. थोड़ा सा डायग्राम बना के आपको कंपेयर करना पड़ेगा. थोड़ी सी थिंकिंग लगेगी. Towards the pole of cell. Question 20. Which one is correct for the start of anaphase of mitosis? तीन चार क्वेश्चन दे दिए उन्होंने माइटोसिस पे एक साथ. फॉर्म ऑफ डीएनए. अच्छा. मैट. Chromosome separated sister chromatid. During anaphase the centromere divides which is why आपके जो chromatids होंगे वो separated होंगे. C और D. DNA is associated with histone protein at all time. कभी भी ऐसा नहीं होगा कि आपके histone protein and your DNA separate. So it is always associated with your histone protein. State of the cell surface membrane, it is intact. Broken apart कब होता है? जब आपका cytokinesis हो रहा होता है. And cytokinesis happens after your mitosis का step. So yes, your state of cell surface membrane will be intact. C is your correct option. The diagram shows chemical structure of four bases. कौन सी bases? ATGC. या AUGC, let's see. Which diagram shows thymine and cytosine? Thymine and cytosine. If you remember the trick, CUT. This was for pyrimidine bases. Pyrimidine bases, and you know pyrimidine bases are smaller bases with a single ring structure. Smaller bases with a single ring structure. So you're going to look for structures with one ring in them. इसमें दो हैं, ये wrong है. अच्छा ये वाली cut वाली track, cytosine, uracil and thymine. So option 2 is correct. Option 3 is correct. So option 4 is wrong क्योंकि आपको इसमें double ring structure नजर आ रहा है. This is your purine base. Same goes for your option 1. Option 1 is also your purine base, larger bases with the double ring structure. And they are adenine and guanine. तो इसमें से कोई एक adenine होगी, कोई एक guanine होगी. Whereas for 2 and 3 इसमें से कोई एक thymine होगी and the other one will be cytosine. The correct option is 2 and 3. Is this clear? I hope. Okay. Question 22. The diagram shows the nucleotide sequence of a small section of the transcribed strand of a gene. Transcribed हुआ है. Translation translated. Right. The table shows the amino acid coded by for 10 mRNA codon. What is the sequence of 4 amino acids in the polypeptide translated from this small section of gene? So आपको gene sequence दिया हुआ है, nucleotide sequence दिया हुआ है. You need to translate it first and then you're going to compare it with your amino acid the sequence. So let's translate. अच्छा, GCGG will be translated into GCC. फिर कॉम्प्लीमेंट्री बेस पेयरिंग हो रही है, बस आपको वही चेंज करना है. CGG and GGC, GCC. CGG is converted into GCC या फिर translated into. Now you need to compare from your table कि कौन सा amino acid is coding for which triplet. GCC ये रहा. Alanine हो गया ये आपका. CGG, CGG ये रहा. Arginine हो गया आपका. Alanine, arginine. If you look at your options, this eliminates A, C and D. B is your correct option, but let's see. GGC. GGC आपको नजर आ रहा है glycine. This is your glycine. GCC is again your alanine. तो पूरा कोड आपका आ गया. Polypeptide sequence आ गया, which is alanine, arginine, glycine and again alanine. Question 23. What does the process of translation require? पहले transcription होती है and then you have the process of your translation. Translation occurs on your ribosome. तो obviously आपको ribosome की need होती है. Translation involves your mRNA. mRNA is the one which is translated. So obviously you need mRNA to start the process of translation. फिर mRNA पे आपका codon, anticodon एक specific structure carry कर रहा होता है. That is carried by your tRNA. And the triplet is coding for your amino acids. So you need amino acids as well. ये चार चीज़ें आपको चाहिए होती है for the process of your translation. Whereas for transcription, you need pre-nucleotide bases, you need DNA, and you need your nucleus and your RNA polymerase as well. ये चार चीज़ें चाहिए होती है for the process of your transcription. Let's see the option. A. DNA. DNA and pre-nucleotides are needed for the process of transcription. तो ऑप्शन ए तो हमारा एलिमिनेट हो गया. डीएनए अगेन ट्रांसक्रिप्शन के लिए चाहिए होता है. एमआरएनए अमाइनो एसिड आरएनए पॉलीमरेज इज नीडेड फॉर द प्रोसेस ऑफ ट्रांसक्रिप्शन. ऑप्शन बी इज आल्सो एलिमिनेटेड. एमआरएनए राइबोसोम्स आरएनए पॉलीमरेज अगेन वही लॉजिक फॉर द प्रोसेस ऑफ ट्रांसक्रिप्शन. वी आर लुकिंग फॉर द प्रोसेस ऑफ ट्रांसलेशन. mRNA, ribosomes, amino acid, and tRNA. This is your correct option. So make sure you remember the process of transcription and translation. The electron micrograph shows a longitudinal section of a part of the stem of a plant. What is the name of the structure labeled X? Look at your options. Minutes. Companion cell, Casparian strip, phloem sieve tube, xylem vessel element. Casparian strip तो ये हो ही नहीं सकती. If you look at your structure, यहाँ पे आपको दिख रहे हैं. Yes. यहाँ पे आपको नजर आ रहा है perforated sieve plate. Perforated sieve plates. फिर आपको यहाँ पे बराबर में नजर आ रहे हैं योर कंपेनियन सेल. So this is obviously your phloem sieve tube element, companion cell. Structure X is your phloem sieve tube. Which substance makes xylem vessel wall impermeable to water? क्या आंसर होगा? Xylem vessel element. Xylem vessel wall में एक चीज होती है that is making it impermeable to water. Yes. C? C तो उसमें होता है, Casparian strip में होता है. Yes, lignin होता है आपके पास. Lignified walls होती हैं in your xylem vessel element. C. Suberin is in your Casparian strip, which is blocking your apoplastic pathway. Casparian strip. तो रिवाइज कर लेना एक बार. Strip. Cellulose is in your cell wall. Lignin is in your xylem vessel element. The following tissue carry an electrical impulse during the cardiac cycle. In which order does the electrical impulse travel during the cardiac cycle? This is again a repeating question. काफी बार हम इसे list कर चुके हैं. So it starts from your sinoatrial node present in your right atrium. Sinoatrial node spreads the impulse throughout the walls of your atria, जिसकी वजह से आपकी जो atrial walls होती हैं, they contract. Your atria contract. So the muscle of the wall of your atria, this is your second. फिर third क्या होता है? ये impulse goes to your atrioventricular node. Atrioventricular node पे जाती है, which is 1. फिर it goes to your Purkinje tissue. Purkinje tissue is present at the apex. Apex या फिर base of your heart, and it facilitates the contraction of your ventricle. Atrioventricular node ensures कि आपके atria पहले contract करें and then your ventricle. It causes a brief delay in the transmission of your electrical impulses. So the order should be 4, 2, 1 and then 3 in the last. Purkinje tissue in the last, which is seen in option 4 to 1 is C. Question 27. The graph shows the changes in the volume of ventricles during a simple cardiac cycle. Acha. Which was correct for the atrioventricular valve at P and at R and the semilunar valve at R? अच्छा, सही है. Atrial systole मतलब आपका atrial contract कर रहा था. Then your ventricular systole happened, जिसमें आपके ventricle contract कर रहे होते हैं. When your ventricle start to contract, a particular valve starts to close. तो यहाँ पे एक particular valve है which is starting to close. Completely close नहीं हुई है but close करना start कर दिया इस valve ने. And that valve is your atrioventricular valve, which includes your mitral and tricuspid valve. Tricuspid valve. तो ये दोनों valve आपकी यहाँ पे close हो रही हैं and they are known as your atrioventricular valve. Diastole. Diastole refers to the relaxation of your ventricles and your atria. यहाँ पे इन्होंने specify नहीं किया हुआ किस चीज़ का diastole हो रहा है. So you're going to assume कि आपको ventricle and atria दोनों relax कर रहा है. And you know जब आपका diastole हो रहा होता है, especially जब दोनों का हो रहा हो, तो all your walls are closed. All walls are closed, which includes your semilunar and your atrioventricular valves दोनों. But semilunar valve पहले close होना start होती हैं. So R पे your semilunar valve closes first. Let's see. Atrioventricular valve at P, they are closing. Semilunar valve at R, they are also closing. Option A is your correct answer. 28. The graph shows how changes in oxygen concentration affect the percentage oxygen saturation of human hemoglobin and cat hemoglobin under normal physiological conditions. The partial pressure of carbon dioxide was kept constant at 5 kilopascal and the temperature was kept constant at 37 degrees Celsius. What conclusion is supported by the graph? If you look at your graph, from your human to your cat, आपका right shift हो रहा है. Bohr effect कौन सा वाला हो रहा है? Right shift वाला हो रहा है. your graph has shifted towards the right if you look at, from the point of view of your human hemoglobin. And right shift में क्या हो रहा होता है? There's an overall reduced affinity for oxygen. Affinity for oxygen. Your hemoglobin molecule is less attracted towards your oxygen molecule, which means जो आपका hemoglobin in cat है, that will be less attracted towards its oxygen. The affinity of cat hemoglobin for oxygen is greater than the affinity of hemoglobin. This is wrong, it is less. Right shift हो रहा होता है, it is less than the affinity. At high partial pressure of oxygen, cat hemoglobin picks up oxygen more easily. This is wrong क्योंकि आपकी जो hemoglobin है that उसकी affinity for oxygen is very less. So it will pick up oxygen less easily. At low partial pressure of oxygen, oxygen is released more easily from cat hemoglobin than from human hemoglobin. This is true. Affinity वैसे कम है, which is why your hemoglobin will give up oxygen very easily in your cat. जब oxygen की concentration low होगी. The shift in the oxygen dissociation curve caused by the Bohr effect is larger for cat hemoglobin than for human hemoglobin. The shift in this we cannot conclude from the graph. This conclusion cannot be made by a graph and is not supported by a graph as well. यही पूछा था हमसे. Which conclusion is supported by the graph? This one is not supported by a graph, which is why this option is wrong. अच्छा, what is the trigger for breathing? किसी को पता है? Why do we need to breathe? आपकी बॉडी में किसकी concentration कम हो जाती है या किसकी concentration ज़्यादा हो जाती है which is causing us to breathe? The trigger for breathing is, ऐसे ही fun fact है, trigger for breathing is actually the concentration of carbon dioxide in your blood. ऑक्सीजन की वजह से आप ब्रीथ इन नहीं कर रहे होते, आपकी कार्बन डाइऑक्साइड कॉन्सेंट्रेशन इंक्रीज हो जाती है इन योर ब्लड प्लाज्मा विच इज व्हाई यू नीड टू ब्रीथ. CO2 concentration in blood plasma. This you'll study in MDCAT in your gas exchange wala chapter. Question 29. Which statement is correct about how oxygen combines with hemoglobin? Combining all four oxygen molecule with hemoglobin does not affect the shape of your hemoglobin. This is wrong. It does affect the shape of your hemoglobin. जैसे ही एक ऑक्सीजन आपके हीमोग्लोबिन पे आके लगता है, it causes a conformational change in your hemoglobin molecule. So it does affect the shape of your hemoglobin molecule. Option A is wrong. One oxygen molecule can combine with each heme group of the hemoglobin molecule. This is correct. The first oxygen molecule to combine with the hemoglobin does affect the shape of your hemoglobin. It causes a conformational change in hemoglobin molecule. The third oxygen molecule to combine with hemoglobin makes it more difficult for fourth oxygen molecule to combine with hemoglobin. According to your course, it does not make it more difficult. But जब आप आगे जाओगे in your MD-CAT या फिर MBBS तब आप पढ़ोगे कि it does make it more difficult for your fourth oxygen molecule to combine with your hemoglobin. But according to your A-level course, the third oxygen molecule to combine with hemoglobin does not make it more difficult. Does not यहाँ पे आएगा, बल्कि it makes it easier. Makes it easier. बिल्कुल opposite है. So the correct option is B obviously. 4 heme group होते हैं. 4 oxygen molecules are able to attach to one hemoglobin molecule. How are alveoli adapted to their function? काफी easy peasy question. What do you think is the answer to this? Yes. Alveoli में सिर्फ squamous epithelium होता है, which is one cell thick. They contain squamous epithelium for a short diffusion distance. A is your correct option. Goblet cell नहीं होते. अगर होते तो आपका diffusion distance affect होता. Diffusion distance आपका increase हो जाता. Ciliated epithelium भी नहीं होते. There is no need क्योंकि mucus ही नहीं बन रहा. So there's no need for a ciliated epithelium. They contain cartilage. Cartilage भी नहीं present होता. It has elastin fiber. Elastin fiber instead. So option D is also incorrect. 31. The diagram shows oxygen diffusing from the space inside an alveolus into the blood through the gaseous exchange surface. What would increase the rate of diffusion of oxygen from the alveolus into the blood? Let's study the diagram, thickness of your alveolar wall and the capillary endothelium. Acha. So oxygen concentration inside the alveolus. Obviously आपका oxygen is entering your alveolus by a gas exchange system जो आपका पूरा respiratory tract होता है. So the concentration of oxygen in your alveolus is very high. High. The concentration of oxygen in your Y जो आपकी walls of your capillary and alveolar wall है इसमें low होता है oxygen concentration ताकि diffusion distance diffusion gradient या concentration gradient develop हो सके. Oxygen concentration in the blood relatively same होती है. It remains same. Increase. अच्छा लेट्स से आपको नहीं पता कि यहाँ पे लो है कि यहाँ पर लो होगी W में लो होगी या Y में लो होगी. You can look या you can predict it from your options as well. W Y इन दोनों में increase नहीं हो सकता. Your oxygen concentration is only high in your alveolus ताकि आपको oxygen या diffusion gradient develop हो सके. This is your correct answer. Is this clear? What will increase the rate of diffusion of oxygen? So obviously alveolus में high होना चाहिए so that your oxygen can enter into your blood. I hope this is clear. What tissues are present in a bronchus? Bronchus and trachea दोनों में similar structure present होते हैं which is your cartilage, your ciliated epithelium and your smooth muscle as well. तीनों चीजें present होती हैं. A is your correct answer. Steep concentration gradient must be maintained for efficient gaseous exchange to occur in the human lung. Which row correctly describes how steep concentration gradient can be maintained? Acha, elastic fibers in the wall of the alveoli recoil when breathing out. So yes, they do recoil when breathing out and stretch when breathing in. Stretch when breathing in. And this is helping in your steep concentration gradient as well. This is tick. Continual supply of deoxygenated blood by the pulmonary artery. This is also true. Your pulmonary artery is carrying deoxygenated blood and अगर continuous supply आता रहेगा तो continuous gas exchange होता रहेगा. So this is also correct. A is your correct option. ये math वाला question है. You should know the formula of percentage increase and percentage decrease. A scientist investigated the effect of an antibiotic on the treatment of cholera. 320 people were divided into two groups. One group was treated with antibiotic while the other group was not given the antibiotic. Both groups were given fluid containing sugar and mineral salt. This is your oral dehydration therapy, ORT. ये आपने पढ़ा भी होगा. The scientists recorded the number of days that each person had diarrhea. The table shows the result. Antibiotic and oral rehydration therapy. Meantime, a person had diarrhea कम था. Whereas अगर आपने सिर्फ ORT दी हुई है, so meantime the person had diarrhea वो ज़्यादा है. What is the percentage decrease in the meantime that a person had diarrhea when they were treated with the antibiotic? So percentage decrease का formula क्या होता है? किसी को पता है? Old value वाला. This is your new value. This is your old value. Yes. Uh, किस तरह होता है? New value minus your old value. Hmm. Divided by your new value. Percentage decrease find out करना है. Multiply by 100. परसेंटेज इंक्रीज भी मांगेंगे आगे क्वेश्चंस में, तो फॉर्मूला बिल्कुल ऑपोजिट हो जाता है. लेट्स सी. Hence we will subtract 5.3 by 3.2 divided by 5.3 multiply by 100. Calculator से निकाल लो. The answer will come out to be 39.6%. कुछ भी नहीं करना था, सिर्फ कैलकुलेशन करनी थी. ओल्ड वैल्यू तब आती है जब आप परसेंटेज इंक्रीज निकाल रहे हो. वो वाला क्वेश्चन भी कर लेते हैं. 20 से skip ये रहा. 36. Scientists studied the multi-drug resistant bacterial infection in children caused by one type of bacteria between 2007 and 2015. The percentage of multi-drug resistant infection rose from 0.2 to 1.5. So 0.2 is your old value. Old value या फिर original value एक ही बात. What was the percentage increase in multi-drug resistant infection between 2007 and 2015? So you're going to subtract your new value from your old value. न्यूमरेटर सेम रहेगा, बस आपका डिनोमिनेटर चेंज हो रहा होता है इन योर परसेंटेज इंक्रीज एंड डिक्रीज. इंक्रीज के लिए यू डिवाइडेड बाय योर ओल्ड वैल्यू जो आपकी पुरानी वैल्यू थी एंड मल्टीप्लाइड बाय 100. तो ओल्ड वैल्यू वाज 0.2, वी आर गोइंग टू डिवाइड इट बाय 0.2. 1.5 माइनस 0.2 डिवाइडेड बाय 0.2 मल्टीप्लाई बाय 100. आंसर आएगा आपका 650%. दिस इज द परसेंटेज इंक्रीज. करेक्ट ऑप्शन इज डी. तो ये दो क्वेश्चंस काफ़ी अनफेयर हैं, मैथ्स के पता नहीं क्यों दिए हैं इन्होंने. बीच में क्वेश्चन 35. हाउ डज द एंटीबायोटिक पेनिसिलिन अफेक्ट द मेटाबॉलिज्म ऑफ अ बैक्टीरियल सेल? सो इट स्टार्ट कब एक्ट करता है? जब आपका सेल वॉल सिंथेसिस हो रहा होता है इन योर बैक्टीरिया. पेनिसिलिन reduces cell growth. This is wrong. It is not affecting the mitosis या cell growth of your bacterial cell, and it is also not affecting the water uptake. Penicillin inhibits the formation of cross-link in the cell wall between peptidoglycan molecule. This is correct. बिल्कुल सही है, सारे पॉइंट इसमें सही हैं. Penicillin inhibits the hydrolysis. No, it is not inhibiting the hydrolysis of peptidoglycan link. It is inhibiting the formation, condensation of your cross-link during cell extension. This is wrong. Penicillin weakens नहीं कर रहा होता है, it inhibits the cell wall by digesting peptidoglycan molecule. डाइजेशन इसके मैकेनिज्म में नहीं होता, पेनिसिलिन के मैकेनिज्म में. सो दिस वाज आल्सो एन इजी क्वेश्चन. देख के आपको पता चल जाता आंसर. ऑल्सो ये दोनों फॉर्मूले याद कर लेना. परसेंटेज डिक्रीज का ये वाला फॉर्मूला है एंड दिस इज द फॉर्मूला फॉर परसेंटेज इंक्रीज. परसेंटेज इंक्रीज. ओके. 37. Whooping cough is a highly infectious disease of the gas exchange system caused by bacterium Bordetella. अजीब सा नाम है. Which method provides protection to infants during whooping cough against whooping cough and reducing the chance of developing this disease later? A short course of more than one type of antibiotic. इसकी वजह से क्या अफेक्ट आएगा? Yes. And more than one type इसी वजह से भी आपकी एंटीबायोटिक रेजिस्टेंस डेवलप हो सकती है. A 6 month course of one type of antibiotic. One type का मसला नहीं है, 6 month. 6 months is too long. आप चांस दे रहे हो बैक्टीरिया को एंटीबायोटिक रेजिस्टेंस डेवलप करने का. So this again is going to cause you antibiotic resistance. Injection of antibodies specific to this virus, bacterium, sorry. Is this the appropriate answer? No. Option D. Antigens होने चाहिए. Very good. Injections of antigen from bacterium for this particular bacteria. So these antigens will be non-self antigen, foreign particle, non-self antigen, जिसकी वजह से your immune response will be triggered. Triggers immune response. Immune response. And फिर आपके memory cells बनेंगे and this will reduce the chance of developing this disease later क्योंकि आपके memory cells बन रहे हैं. Trigger immune response. Correct option is D. The electron micrograph shows a type of blood cell. What can be concluded from the electron micrograph? If you look at the structure, इसके अंदर काफी सारे एंडोप्लाज्मिक रेटिकुलम है. रफ एंडोप्लाज्मिक रेटिकुलम लग रहा है क्योंकि it looks granulated. अच्छा हाँ, it is granulated. If you look at it, पता नहीं कितना जूम इन हो सकता है. एक मिनट. So if you look at it, थोड़ा सा granulated लग रहा है. Hence we can say कि in a way this is your rough endoplasmic reticulum. और कोई सेल स्ट्रक्चर दिख रहा है? Hmm, ऐसा कोई पर्टिकुलर नहीं नजर आ रहा. So this is your endoplasmic reticulum. रफ लग रहा है. Let's see the option. ग्रेनी नजर आ रहा है, which is why I am concluding कि ये rough endoplasmic reticulum होगा. What can be concluded from the electron micrograph? The cell secretes products that are toxic to pathogen. ये आपका गोल्गी बॉडी करेगा in the form of lysosome. And Golgi body हमें अपने सेल में नजर नहीं आ रहा. So this option is wrong. The cell synthesizes a large quantity of protein. ये हो सकता है a function of your endoplasmic reticulum. The cell synthesizes large quantities of antigen. This is again wrong. The cell digests pathogenic bacteria. So hydrolytic enzymes and those come from your Golgi body. This is again wrong. This is the only possible option. And ये आपका function होता है of your endoplasmic reticulum. Why is mitosis important in the immune response? It allows B lymphocyte to produce plasma cell. पहला ऑप्शन ही करेक्ट है आपका. It allows B lymphocyte to synthesize antibody. Antibody come from your plasma cell. Plasma cell come from your B lymphocyte. तो ये थोड़ा सा ज़्यादा आगे निकल गए. इसमें आप कर सकते हो BCQ. And the best possible answer is plasma cell. It allows neutrophil to carry out phagocytosis. This is not happening through the process of mitosis. इसमें mitosis involved नहीं है, इसमें phagocytosis involved है. It allows T lymphocyte to recognize foreign antigen. So cell recognition is also not an example of mitosis. यहाँ पे cell recognition हो रही है. Recognition. Your correct option is A. Plasma cells are producing antibody, and production of antibody is not an example of mitosis. 40 question 40. What is fused with B lymphocyte to form a hybridoma in monoclonal antibody production? So hybridoma cell कैसे बनते हैं? When your plasma cell fuses with your myeloma cell. Myeloma cell are a type of cancer cell या फिर tumor cell. So the correct option is D, myeloma cell. B lymphocyte is giving you your plasma cell. Plasma cell. तो कभी कभी ऑप्शन में instead of writing plasma cell they do write B lymphocyte as well. And ये हमने एक past paper में देखा भी था. So this was it for today's paper. See you tomorrow inshallah.